Natural Selection, Selective Breeding, and the Evolution of Resistance of Honeybees, Apis mellifera against Varroa, by Jacques J. M. van Alphen and BartJan Fernhout. Abstract. We examine evidence for natural selection resulting in Apis mellifera becoming tolerant or resistant to Varroa mites in different bee populations. We discuss traits implicated in Varroa resistance and how they can be measured. We show that some of the measurements used are ambiguous, as they measure a combination of traits. In addition to behavioral traits, such as removal of infested pupae, grooming to remove mites from bees or larval odors, small colony size, frequent swarming, and smaller brood cell size may also help to reduce reproductive rates of Varroa. Finally, bees may be tolerant of high Varroa infections when they are resistant or tolerant to viruses implicated in colony collapse. We provide evidence that honeybees are an extremely outbreeding species. Mating structure is important for how natural selection operates. Evidence for successful natural selection of resistant traits against Varroa comes from South Africa and from Africanized honeybees in South America. Initially, Varroa was present in high densities and killed about 30% of the colonies, but soon after it spread, numbers per hive decreased and colonies survived without treatment. This shows that natural selection can result in resistance in large panmictic populations when a large proportion of the population survives the initial Varroa invasion. Natural selection in Europe and North America has not resulted in large-scale resistance. Upon arrival of Varroa, the frequency of traits to counter mites and associated viruses in European honeybees was low. This forced beekeepers to protect bees by chemical treatment, hampering natural selection. In a Swedish experiment on natural selection in an isolated mating population, only 7% of the colonies survived, resulting in strong inbreeding. Other experiments with untreated surviving colonies failed because outbreeding counteracted the effects of selection. If loss of genetic variation is prevented, colony-level selection in close mating populations can proceed more easily, as natural selection is not counteracted by the dispersal of resistant genes. In large panmictic populations, selective breeding can be used to increase the level of resistance to a threshold level at which natural selection can be expected to take over. Varroa destructor, Anderson and Truman 2000, is an external parasitic mite of honeybees that shifted from its original host, Apis cerana, the Asian hive bee, to Apis mellifera, the European honeybee. In the mid-1800s, settlers from western Russia carried Apis mellifera to the far southeastern corner of Russia, into an area where Apis cerana occurs naturally. Contact between the two species resulted in the introduction of Varroa to Apis mellifera. The Varroa mite arrived in Moscow during the 1950s with honeybees from the east. The parasite spread rapidly and colonized Western Europe and North America in the early 1980s. And since its invasion from Russia, Varroa has been the major mortality factor of honeybees. Varroa mites are vectors of several bee viruses, and at high mite densities these viruses cause colony collapse. While it does not cause colony mortality in its original host Apis cerana, it became and remains devastating to apiculture and to natural populations of Apis mellifera in Europe and feral populations in North America. One reason why Varroa is so virulent on Apis mellifera is that it can breed in a worker brood and so achieve long reproductive season, while in Apis cerana, pupae from worker cells with reproducing mites are always removed and breeding is restricted to the short season during which drones are produced. Varroa has been present in European and American Apis mellifera populations for almost 40 years, and, as an important mortality factor, it presumably exerts strong natural selection for resistance in these populations. We call a bee colony resistant when it is able to limit the population size of Varroa to a density that does not cause mortality. In Western Europe and North America, hives are frequently treated with acaricides, natural acids, or essential oils to control Varroa, and Varroa reproduction is disrupted by other apicultural measures. Moreover, a large proportion of the hives are regularly requeened. These practices are thought to hamper natural selection for resistance against Varroa. However, not all beekeepers treat their colonies against Varroa, and in Europe, where Apis mellifera is native, wild colonies of honeybees were not uncommon when Varroa arrived. Likewise, populations of feral colonies of honeybees in North America are exposed to natural selection. Evidence for small-scale natural selection from some of these untreated colonies provides insight into why natural selection in the European honeybee has not resulted in population-wide resistance. Traits contributing to Varroa resistance in Apis cerana. The mite stably coexists with its original host. Apis cerana workers prevent the growth of Varroa destructor populations by different behavioral traits known as grooming, uncapping and removing, and entombing. Apis cerana bees groom themselves, orogrooming, and also perform grooming dances to recruit nestmates to engage in social grooming, allogrooming. This often results in the removal of phoretic adult mites and inflicts significant mortality among them. The uncapping of worker cells with reproducing Varroa and the subsequent removal of the parasitized pupae result in the removal of mite offspring before they have been able to reproduce. This is an important factor in preventing the mite population to grow to harmful densities. Of Varroa females that enter a worker cell, 90% do not reproduce, which could be caused by a so-called brood effect, i.e., the suppression of Varroa reproduction by the brood. The few that do lay eggs fail because of the uncapping and removing behavior. In Apis cerana, Varroa only reproduces successfully in drone cells. In drone cells that have been colonized by 2 or more adult females, the host often dies. Apis cerana workers leave the dead drone brood capped, thus entombing the reproducing parasites and their offspring and causing mortality of up to 25% among the mites. Drones are produced during a relatively short season, allowing the mites to produce only 3 to 5 generations per year. This is one of the reasons why Varroa is an innocuous parasite of Apis cerana. Traits for Varroa resistance in Apis mellifera. 3 of the traits that provide resistance against Varroa in Apis cerana are also present in European Apis mellifera populations, albeit in low frequency. The uncapping of Varroa-infected cells and subsequent removal of parasitized pupae, as well as orogrooming and allogrooming. The uncapping of Varroa-infected cells and subsequent removal of parasitized pupae, as well as auto and allo grooming. Another trait that might confer resistance against Varroa is the brood effect. The mechanism for the suppression of mite reproduction is a change in a chemical signal issued by a developing worker larvae that, if unaltered, would be used by Varroa mites as a signal to initiate reproduction. Entombing has not been observed in Apis mellifera. We first review the evidence for the traits that confer or are thought to confer resistance. We show that evidence cited in support of particular resistance trait could sometimes also be produced by one of the other traits. Next, we review the evidence that natural selection has resulted in resistance against Varroa in Apis mellifera in Africa, Europe, and the Americas, and the roles played by the different resistant traits. Uncapping of Varroa-infested cells and removal of parasitized pupae. Hygienic behavior. Hygienic behavior was discovered by Park and co-workers. They observed that bees in colonies that appeared to be resistant against American foul brood removed larvae from cells inoculated with the disease. Their results indicated that the behavior was heritable. Woodrow and Holst labeled this activity hygienic behavior and provided further evidence that resistance to American foul brood in a honeybee colony consists in its ability to detect and remove diseased brood before causative organism, Bacillus larvae, reaches the infectious spore stage in the diseased larvae. Rothenbuehler studied the genetics of hygienic behavior and provided evidence that it is compatible with two recessive genes coding for the behavior. Later research found evidence that more genes are involved, which is not surprising for a behavioral trait. Indeed, Harper et al. Found 73 candidate genes associated with hygienic behavior. Most of those genes play roles in regulating the expression of genes involved in neuronal development, which could explain why hygienic bees are more receptive to olfactory signals associated with dead brood. Woodrow and States already suggested that hygienic behavior was not specific to American foul brood. Open quotation, "It is likely that the removal of diseased brood is a common behavior of bees." Close quotation. Indeed, subsequent studies showed that adult bees from some colonies also removed larvae infected with chalk brood from the cells within 24 hours, and that this behavior also plays an important role in the resistance of bees against this disease. Based on the belief that the hygienic response to diseased and dead brood is general, a test for hygienic behavior using freeze-kill brood was designed. The belief that the hygienic behavior is a general response to diseased and dead brood also led to the hypothesis that hygienic behavior could play a role in resistance against Varroa. This hypothesis was tested by Boking and Drescher and Spivak. Boking and Drescher found a positive correlation, open parenthesis, R equals 0.74, close parenthesis, between the removal of brood infested with 2 mites per cell and the removal of freeze-kill brood, suggesting that the hygienic response to Varroa-infected cells can at least, in part, be explained by the general response of hygienic bees to dead and diseased larvae. Spivak repeated and extended these experiments with similar, albeit variable, results. Hence, hygienic bees selected for removing dead larvae show a higher incidence of removing larvae from cells infested with Varroa than non-hygienic control bees. However, in Boking and Drescher's study, this only explained 55% of the variance. Danke et al. Also found only a weak relation between the removal of dead brood and VSH behavior. Likewise, Boking et al. Showed that the rate of removal of pin-killed larvae correlates with the rate of removal of Varroa-infested cells, but this accounted for only 37% of the variance. They termed the removal of larvae from Varroa-infested cells as, open quotation, Varroa-specific hygiene, close quotation, and were first in recognizing the genetic background of bees removing pupae from Varroa-infested cells is different from bees that only remove killed pupae. Varroa-sensitive hygiene. Evidently, apart from general hygienic behavior, other traits must be involved in the removal of pupae from Varroa-infested cells. The cues bees use to detect and remove frozen or pin-killed pupae are not necessarily the same as those used to detect and remove mite-infested pupae. Harbo and Huppengarner looked for heritable resistance of honeybees without limiting themselves to hygienic behavior. As natural mating of bee queens results in queens inseminated by multiple males, they used queens artificially inseminated with sperm of only a single male to ensure the resistance characteristics could be strongly expressed by a whole colony. Experimental colonies were infested with equal densities of mites, and 63 or 70 days later, the mite population size in the colonies was determined. In addition, They measured the incidence of hygienic behavior using sections of frozen brood, determined the proportion of damaged fallen mites as a measure for grooming behavior, the incidence of non-reproduction of mites that were found inside capped cells, and duration of the cap period as a measure for developmental time. Of these traits, only the percentage non-reproduction showed a negative correlation with the increase in mite population that explained 53% of the variance. They concluded that suppressed mite reproduction, SMR, could be an important resistance trait against Varroa. In a later study, Harbaugh and Harris measured the heritability of potential traits for resistance and found that suppression of mite reproduction, hygienic behavior, and proportion of mites in brood cells were candidates for selection due to high heritability. They then started a program for the selection of bees with the ability to suppress reproduction in the mites that after about 5 generations of selection produced colonies that had less than 6% of their mites classified as reproductive in worker cells. Abraham and Spivak hypothesized that the selective removal of brood infested with reproductive mites could explain the observed decrease in percentage of reproductive mites. Harbaugh and Harris tested this hypothesis. They placed frames with newly capped worker brood in SMR and in control colonies and counted Varroa-infested cells and measured the proportion of reproducing mites after 7 to 9 days. They found that in SMR colonies, the percentage of infested cells had been reduced from 22 to 9%, and the percentage of infested cells containing reproducing mites had decreased from 71 to 20%. The number of cells containing non-reproducing mites was not different between SMR and control colonies. These results show that in the SMR colonies, cells containing reproducing mites had disappeared and cells containing non-reproducing mites had been left untouched. This is the evidence that SMR bees show hygienic behavior preferentially to cells with reproducing mites. Hence, this particular form of hygienic behavior is causing an increase in the percentage of non-reproducing mites by reducing the proportion of reproducing mites. Harbaugh and Harris et al., and Kim et al. Confirmed these findings. Harris renamed SMR and called the behavior of uncapping cells containing reproducing Varroa and the removal of pupae from such cells Varroa sensitive hygiene, VSH, a process that results in the removal of mite offspring before they are able to reproduce successfully, thus interrupting the reproductive cycle of the Varroa. VSH behavior is a heritable trait that responds well to selection. Bees with VSH recognize cells containing reproducing Varroa. There are indications that at least 2 major genes with additive effects are involved. Saruda et al. Did a Qtl analysis for VSH and found 2 QTLs, one on chromosome 9 and one on chromosome 1, together explaining only 10% of the variance. On average, individuals that were homozygous for the VSH allele were more likely to be individuals who were observed exhibiting VSH. Spotter et al. Found 6 candidate genes, and Scanopico et al. Found 5 genes associated with VSH behavior. It is likely that some of the genes involved in hygienic behavior also play a role in VSH. The proportion of workers in a colony expressing VSH behavior is positively correlated with the proportion of non-reproducing mites in the brood. This is because VSH bees preferentially attack cells with reproducing mites. A low frequency of VSH behavior must be present in almost every population of European honeybees in Europe and North America because it has been found when looked for, e.g., low VSH in commercial control colonies. Recapping. In experiments to measure VSH, it has been often observed that pupae infested with Varroa that had been uncapped were recapped without the host pupae being injured. Although the foundress mite may escape an uncapped brood cell before it is recapped, she usually remains within the cell. Brood exposed for 1 week to bees selected for VSH often have high mean percentages, greater than 30%, of recapped brood cells, and some colonies may have greater than 90% of all brood recapped. Most of these recapped cells are not infested by Varroa, but about 20% of recapped cells contain a mite. Martin et al. Consider recapping as the repair of cells erroneously opened by VSH bees. Harris et al. Suggested that it is possible that hygienic uncapping followed by recapping of brood cells could inhibit or alter mite reproduction. This would be an alternative explanation for the increased percentage of non-reproductive mites found by Harbaugh and Harris, who did not discriminate between normally capped and recapped cells. This hypothesis was tested by Harris et al. They found that the frequency of pupae with remaining fertile mites in normally capped brood cells for control bee was 10 times that found for VSH bees, confirming Harbaugh and Harris' conclusion that VSH bees preferentially target cells with reproducing mites. The total number of offspring for fertile mites did not vary between normal and recapped cells, indicating that offspring were not removed before cells were recapped. Mortality of mite offspring was significantly higher in recapped cells than in normally capped cells and contributed to decreased reproduction by the mites. Harris et al. Consider recapping to be part of VSH behavior. However, in the colonies labelled as VSH, not all bees exhibited VSH behavior, as part of the workers were from patrolines not expressing VSH. Therefore, it could also have been non-VSH bees that recapped the cells opened by VSH bees. Evidence that non-hygienic bees recap cells comes from Spivak and Gilliam. When they added young, non-hygienic bees to hygienic colonies, hygienic behavior was suppressed. In a different experiment, they showed that non-hygienic bees tended to recap partially uncapped cells containing dead brood, whereas hygienic bees never recap those cells. De Guzman et al. Found higher recapping rates in Italian bees with low rates of VSH behavior than in the resistant Russian honeybees that displayed high rates of VSH. More evidence comes from Boking and Spivak, who found that bees from pre-selected non-hygienic colonies tend to recap partially uncapped cells that contain freeze-kill brood, and from Arathi et al., who demonstrated that in mixed colonies, as compared to a colony of hygienic bees, a higher proportion of uncapped cells were subsequently recapped, resulting in delayed removal of dead brood. Hence, recapping by non-hygienic bees appears to counteract the activity of the hygienic workers and probably reduces the efficacy of hygienic behavior against Varroa. More information is needed to know how this depends on the ratio between non-hygienic and VSH bees in a colony. Measuring Varroa-sensitive hygiene. Many studies have used frozen or pin-killed brood to assess the rate of removal of pupae infested with Varroa. These assays measure hygienic behavior which, as we have seen, correlates only partly with VSH behavior. Therefore, VSH should be measured using brood infested with Varroa. Grooming, auto and allo grooming. As in other insects, adult bees clean their bodies by grooming themselves, auto-grooming. As mentioned above, they may also perform a grooming dance to solicit grooming by other bees. Grooming by other bees is called allo grooming. This behavior has been described in detail by Land and Seeley. Auto and allo grooming are performed by all honeybees to remove dust or pollen. Auto and allo grooming in response to the presence of a Varroa mite play important roles in the resistance of Apis cerana against Varroa. In Apis cerana hives, 73.8% of fallen mites exhibited damage. The evidence is based on direct observation of the grooming behavior in combination with data on the rate of successful mite removal from bees, the percentage of mites dropping to the bottom board, and the percentage of damaged mites. It points to a direct relationship between grooming behavior and the falling of mites to the bottom board. The Varroa mites in the Peng et al. Publication originated from Apis mellifera. This may have resulted in a higher percentage of damaged mites than if the Apis thorana bees had been tested with mites from conspecifics. Incidence of grooming against Varroa in Apis mellifera. Grooming behavior against Varroa in Apis mellifera is expressed at a lower frequency and intensity than in Apis thorana. Depending on subspecies or on differences between colonies of the same subspecies, Apis mellifera adults vary in response to being mounted by a Varroa mite. They may or may not react by autogrooming or by performing the invitation to grooming dance. Apis mellifera bees from colonies that are resistant to Varroa show more vigorous and more effective grooming responses, but good data on the relation between grooming and mite mortality are not available. Measuring the incidence of grooming behavior. Grooming against Varroa mites has been associated with higher proportions of mutilated mites falling from bees in colonies, and the proportion of mutilated mites was associated with lower infestation levels. Moos-Beckhofer found a significant negative correlation between the proportion of dark damaged fallen mites and brood infestation rates. Apis mellifera colonies with the lowest rate of mite population growth showed more grooming behavior, more mites falling to the high floor, higher proportions of fallen chewed mites, and reduced infestation levels of adult bees. These observations have been the basis for using the proportion of damaged mites as a measure for grooming behavior, and the belief that it may be a useful parameter in selecting for Varroa resistance. The proportion of damaged mites fallen to the high floor varies greatly between colonies and between subspecies. Colonies of Apis mellifera linguistica showed an average mite damage rate of 5.75%. In contrast, Rosenkrantz et al., working with Apis mellifera linguistica and Apis mellifera carnica, recorded mite damage rates averaging 45%, open parenthesis 44 to 62%, close parenthesis, while Africanized Apis mellifera damaged 38.5%. Rutner and Hanel provided evidence for active defense of some surviving colonies of Apis mellifera carnica against phoretic Varroa, based on the observation of fallen mites with damaged legs and cuticle of the dorsal shield, or idiosoma. Later it was reported that these strong Apis mellifera carnica hives eventually achieved a damage rate of 93%. Apis mellifera has not been as well studied as the above-mentioned subspecies, but a Polish population of Apis mellifera bees was much more reactive to Varroa mites than bees from local populations of Apis mellifera carnica and Apis mellifera caucotica. 98% showed some response to contact with a mite. The question is whether the variation in the proportion of damaged mites reflects heritable variation in grooming behavior against Varroa mites. Moos-Beckhofer reported that 53.7% of the mites that had fallen to the bottom in the AM carnica colonies he studied were light-coloured young females, 27.1% of which were damaged. Such light-coloured females may well have originated from cells after eclosion of a young parasitized worker bee. However, they could as well have originated from cells opened by workers showing VSH behavior and have been removed with the parasitized pupa or during tidying up of the cell after removal of a pupa. Supporting evidence for the latter scenario comes from Hoffman, who assessed damage rates in fallen mites in relation to the development of the brood nest. When no brood was emerging, the damage rate was 10.2%, but was significantly higher at 16.7% with emerging brood, when the multiple injury rate of mites was also higher. Lobb and Martin estimated that around 50% of fallen dead mites die within seal brood cells, the rest mainly shortly after emergence. Martin reported that numbers of fallen mites increased by a factor of 6, or 7 to 15, when Apis mellifera brood was emerging, compared to when it was not. Rosenkrantz et al. Monitored the proportion of damaged mites in the floor debris of Apis mellifera hives, with and without emerging brood, and when Varroa-sensitive hygiene was stimulated by the insertion of newly killed, but otherwise intact Varroa mites. They found that dead mites removed from brood cells by the bees were damaged to a similar extent as those removed by grooming, which was maximal when brood was emerging. Likewise, Irene et al. Found that mite fall was positively correlated with VSH behavior in Russian honeybees. Hence, a considerable proportion of the fallen and the damaged mites could result from VSH or cell cleaning activities. Moreover, dead mites could also have been damaged by other organisms scavenging in the hive debris, like wax moth larvae or ants. Andino and Hunt showed that grooming activity does correlate with the proportion of fallen damaged mites, but only explains 23% of the variance. Thus, the percentage of damaged mites is not a reliable indicator of the extent of successful grooming taking place. Other methods to measure and quantify grooming behavior. There are additional methods to assess grooming behavior in response to Varroa. Direct records of grooming in observation hives, bioassays with frame with several hundred of bees, and bioassays with individual bees or small groups of bees in petri dishes. Of these, direct observations using an observation hive resembles a natural setting. Also, it measures grooming effort and its success directly, avoiding the problems of interpretation associated with mitefall data. The method is unfortunately time-consuming, which has been the reason for the more common use of simple bioassays with isolated bees in petri dishes. These bioassays are useful to measure differences in the rate of grooming behavior between different bee populations. These do not, however, provide a measure of mite mortality. Mites dislodged from the bee's body by grooming fall on the bottom of the petri dish and can remount the bee. On a vertical frame, mites dislodged from bees may fall to the bottom of the hive. Simple bioassays are also not suitable to measure allo-grooming frequency. The frequency of mite injuries showed no correlation with bees' auto-grooming capacity. This suggests that most injuries to mites are likely to be caused by an activity other than auto-grooming per se, e.g., during allo-grooming, hunting of non-phoretic individuals, or by VSH. Therefore, data obtained by observing isolated bees in petri dishes, as has been proposed as an assay for grooming, may be an or weakly related to mitefall and percentage damaged mites in a colony. Andino and Hunt's bioassay with a single frame still uses mitefall and proportion of damaged mites to assess effective grooming behavior. However, they exclude VSH as a source of mitefall by using a frame with only nectar and pollen and no brood, and thus most fallen mites must have been removed from the adult bees by grooming. Their assay could be a suitable one to measure grooming behavior, in particular when a variant with a frame with open brood is used. It could be used in a selection program to increase grooming. The heritability of grooming behavior. Buchler et al. Selected for an increased proportion of damaged fallen mites. Although they found an increase after several generations, the estimated heritability was low, H2 less than 0.15. They concluded that the heritability was too low to justify the laborious sample collection and processing in a large-scale selection program. Stanimirovitz also measured heritability as a percentage damaged fallen mites and found a variable but rather low heritability, 0.16, less than H2 less than 0.42. Moretto et al., using an observation hive to assess the importance of grooming by direct observation, found an estimated heritability of H2 equals 0.71 plus minus 0.41. Buchler et al. Concluded, against empirical evidence, that the proportion of mutilated mites in the debris of a colony can be used as an indicator of grooming success under field conditions. As only a fraction of the mutilated mites is due to grooming behavior, their conclusion that the heritability of grooming is too low to justify a selection program is not supported by proper empirical data. When properly measured, heritability of grooming behavior could well be high enough for a successful selection program to increase grooming. Importance of grooming as a defense against Varroa. Grooming against phoretic Varroa mites is the only defence that bees have during the long period in winter when there is no brood nest. Honeybee colonies with a high frequency of effective anti-Varroa grooming could in this way reduce the weakening and mortality of the worker bees during winter and reduce the parasite population to a low level before the new reproductive season starts. Including anti-Varroa grooming behavior in a selection program for Varroa resistance could therefore be considered, although we do not know if anti-Varroa grooming behavior is an essential trait for resistance against the mite. And dino-enhanced bioassay to estimate grooming frequency would be a good compromise to minimize the laborious sample collection and processing. Non-reproducing Varroa females. Brood effects. The hypothesis. Many studies have found evidence that not all female Varroa that enter a cell reproduce. There are 4 hypotheses that can explain non-reproduction of foundress mites. 1. As described above, Harbaugh and Harris initially interpreted the increased proportion of non-reproduction in the lines selected for Varroa resistance as suppression of mite reproduction by the pupae. It turned out that the removal of pupae with reproducing mites by bees from these lines caused most of the increase in the proportion of non-reproducing mites. 2. However, non-reproduction can also be a trait of Varroa. On its original host, Apis cerana, a large proportion of adult female mites enter work cells but do not reproduce, and Boot et al. Asked why they would enter the worker brood cells if they do not reproduce there. Apparently, reproduction is not the only reason for mites to invade a brood cell. They may invade worker cells of Apis cerana to hide in safety from the grooming behavior of adult bees and so survive periods without drone brood. In Apis cerana, the drones are produced during only 3 to 4 months. With Apis cerana drone post-capping development times of 13.5 to 14 days, a single fertile mite would have only approximately 3 to 5 reproductive cycles per year. This means that adult mites may spend 8 to 9 months of the year without opportunity to reproduce, and would be exposed to grooming behavior during this time if they would spend it as phoretic mites. 3. A third hypothesis is that there is a constraint on reproduction in these mites. This could either be because they have not been inseminated or because of other reproductive problems. Martin et al. Summarized the published evidence that in Europe, 6 to 24% of adult Varroa females enter cells but do not reproduce. In an experiment, he showed that 8 to 20% of male Varroa offspring in worker cells and 10% in drone cells died before being able to mate. Unfertilized females of Varroa only produce male offspring. A recent finding is that unfertilized females may sometimes mate with their own son and then be able to produce daughters during a second reproductive cycle. Yet, unfertilized females can explain a large part of the observed non-reproduction. Other studies found similar results. Open parenthesis, 12% non-reproduction, 11 to 17% only male offspring, close parenthesis. Constrained females may explain most of the non-reproduction of Varroa observed in mite-susceptible colonies. 4. A fourth hypothesis deals with Varroa-tolerant or resistant bees. Kamazine compared Varroa reproduction on European honeybee and Africanized honeybee. He introduced combs of Africanized and European honeybee larvae into mite-infested Africanized bee colonies. In European honeybees, 75% of infested brood cells had female mites that reproduced, while in Africanized honeybees this was only 49%. As only the origin of the brood was different in his experiment, a factor in the brood apparently affected the reproductive success. Harbaugh and Harris found that the increase in the proportion of non-reproducing mites by VSH could not explain all non-reproduction. A second trait contributed to this reduction. A genetically based factor from the brood produced by VSH queens reduces mite reproduction. Thus, in these selection lines, VSH is not the only mechanism resulting in a reduction of mite reproduction. 3 studies now provide evidence that the mites do not always reproduce after entering a drone cell, and that there are genetic differences between the drones on which mites reproduce and drone brood on which they do not. The inhibition of Varroa's reproduction by infested pupae, open parenthesis, i.e. brood effect, close parenthesis, is a shared trait of many Varroa resistants. The inhibition of Varroa's reproduction by infested pupae, i.e. a brood effect, is a shared trait of many Varroa-resistant Apis mellifera populations across the globe, as well as in the original host, Apis cerana. The mechanism causing non-reproduction. Stage-specific signals of the host larvae initiate and disrupt Varroa reproduction. Chamazine suggests a lower level of juvenile hormone, open parenthesis equals JH, close parenthesis, production in Africanized bee larvae, as hypothesis to explain the lower proportion of reproduction of Varroa in Africanized honeybees. The available evidence published at the time showed that JH titer indeed affects reproduction in Varroa. It was hypothesized that this hormone could also regulate oogenesis in Varroa, and in addition, that host-derived JH could be responsible for initiation of reproduction. When more sensitive techniques were available to measure JH titers, follow-up studies could not confirm these results. More recently, Conlon et al. Found evidence that a gene from the ecdysone pathway could be involved in the suppression of reproduction of Varroa. Varroa requires ecdysone and pupal proteins to initiate vitellogenesis but lacks the genes for ecdysone synthesis. Other chemical signals than ecdysone emitting from the larva could be involved in inducing the Varroa mite to enter a cell, or inducing Varroa to start reproduction. Understanding the underlying physiological process that interfere with the crosstalk between the mite and the host larva will be fundamental to comprehend exactly how the brood effect works. Villa et al. Tried to increase suppression of Varroa reproduction by selection for the brood effect. They found a significant response during the first 2 generations of selection, but the difference between selected colonies and control colonies disappeared in successive generations. A possible explanation for this finding is that adaptation of mites to host cues occurs in these experiments. Other brood effects. Varroa-infested brood from hygienic colonies was more likely to be removed than brood of unselected colonies in cross-fostered brood experiments, showing that a factor in brood is involved in VSH behavior and mediates Varroa resistance. Hence, brood effects and VSH are partly interdependent. Signal production by parasitized pupae and perception of the adults can both play roles in the detection of infested cells. Brood cell size. The natural cell sizes of European honeybees, Apis mellifera, were smaller than nowadays found in most beehives. Beekeepers wanted more productive bees and started to use foundation with larger cell sizes, as this was believed to improve colony performance. Erickson et al., however, suggested that the natural smaller cell size might be advantageous for a number of reasons, including resistance against Varroa. Their hypothesis followed from the observation that Africanized honeybees build small cells, diameter 4.5 to 4.8 millimeters, in comparison with those of European bees, diameter 5.1 to 5.5 millimeters, and that Varroa has a much lower reproductive success in Africanized bees. Independent tests of Erickson et al.'s hypothesis using a variety of different experimental designs and a variety of criteria to judge the hypothesis have produced variable results. Heath provides a review of these studies. He cites 15 studies of which 5 provide support for the hypothesis by Erickson et al. Hypothesis. One hypothesis to explain a lower reproductive success of Varroa in smaller brood cells is that bees in smaller cells have a shorter developmental time, leaving less time for reproduction of Varroa. A second hypothesis is that immature mites may experience difficulty developing in small cells due to lack of space, impeding movement of the mites and possibly causing an increase in mortality of mother mites and offspring. Population growth of Varroa. Martin and Krieger found evidence in support of this hypothesis when they compared the number of offspring per cycle of Varroa in brood of Apis mellifera scutellata with that in brood of the larger Apis mellifera campensis bees in Apis mellifera scutellata cells. Seeley and Griffin compared bees of the same origin that were either placed on frames with small 4.8 mm or large 5.4 mm cells. They measured population development of Varroa once a month, from mid-June to mid-October, and did not find differences in population growth of the mites. They attributed the lack of differences to the small difference in available space between the two types of cells, caused by differences in the size of bees developing in small and large cells. Likewise, no larger Varroa populations were found in hives with large cells. Number of offspring per cycle. Although in choice experiments smaller brood cells have a smaller probability of being colonized by Varroa foundresses, no effect of cell size on the number of female Varroa offspring was found. Hence, neither the hypothesis that shorter developmental time of bees in small cells results in slower population growth in Varroa, nor for the hypothesis that lack of space in small cells restricts Varroa reproduction is supported by experimental results. All the evidence that small cells reduce Varroa populations growth come from experiments with African or Africanized bees. Maggie et al. Found that Varroa was more often non-reproductive in cells with a smaller width. This suggests that the effect is caused by an interaction between cell size and another resistant trait, as explained in the following paragraph. Interaction between cell size and VSH. A possible explanation for the variable outcome of studies on small cell size is an interaction between cell size and VSH behavior. Smaller cells may enhance brood signaling, i.e., in smaller cells suppression of Varroa reproduction by the worker larva, or recognition of cells with reproducing Varroa could be easier. Evidence for this hypothesis comes from reference 122. Some of the selection lines that were used did not show the VSH trait and provide evidence that cell size per se does not influence Varroa population growth. In selection lines with the VSH trait, the rate of the VSH cleaning behavior was higher on the smaller cell size. This confirms earlier findings, reference 123. Hence, the variable outcome of studies on small cell size could be caused by variation in VSH behavior of the bees used in the different studies. Resistance and tolerance against the Varroa-associated viruses. An important reason why Varroa-infested colonies collapse is that Varroa activates an infection by deformed winged virus, DWV, and increases the titer of other viruses such as sac brood virus and black queen cell viruses. The latter two have also been implicated in Varroa-induced bee mortality. These viruses have been associated with the deaths of millions of European honeybee colonies across the world. Bees in the absence of such viruses can sustain much larger Varroa populations before collapse than bees exposed to the virulent forms of these viruses, as was observed in South Africa. The interaction of Varroa with the DWV honeybee interaction somehow results in the virus becoming much more virulent, although this has not happened in the honeybee population on the island of Fernando de Noronha in Brazil. Varroa surviving bees in the Swindon Honeybee Conservation Project also were predominantly infected with an avirulent type of DWV. Vertical transmission favours the evolution of lower virulence. As vertical transmission is more frequent in closed populations, It is expected that dominance of the avirulent form of DWV is found on islands as Fernando de Noronha or otherwise isolated populations like Gotland, the Arnott Forest, and Swindon. In addition to lower virulence of the parasite, increased tolerance resistance of the host is also favoured in a system with predominantly vertical transmission. Evidence for the role of virus tolerance in the Gotland population comes from reference 130 to 132 and reference 133. Likewise, Apis mellifera scutellata seems resistant or tolerant to DWV. The evolution of lower virulence in DWV and other viruses is also favoured by VSH. The hygienic bees preferentially target pupae that have been damaged by a virulent form of the virus, and so can be instrumental in making a less virulent type dominant. Bee life history traits that may hamper Varroa population growth. Colony size and swarming frequency are life history traits of bees that affect population growth of Varroa. Although these traits are to some extent heritable, they are largely determined by environmental factors like nest size and food abundance. The mechanisms whereby smaller and more frequently swarming colonies have greater resistance to Varroa destructor include having relatively few brood cells, especially drone brood, which limits reproductive possibilities for the mites. Frequent swarming also helps control the mites because a swarming event exports about 35% of a colony's mites. Furthermore, swarming temporarily deprives the mites of brood, and its absence disrupts the mites' reproduction and increases their exposure to grooming. Feral bees in Arnott Forest, New York, were infested with Varroa mites, but had a stable population size with established colonies having a lifespan of 5 to 6 years. Seeley and Loftus et al. Tested the hypothesis that persistence of these wild colonies is at least partly due to their habitat of nesting in small cavities and swarming frequently by comparing colonies in small and large hives. These results confirm that smaller nest cavities and more frequent swarming of wild colonies contribute to their persistence without mite treatments. In addition, Seeley and Smith showed that crowding of beehives in apiaries increased Varroa transmission between colonies. They concluded that the scattered distribution of wild colonies makes them less exposed to horizontal infection from other colonies by drifting and robbing, as occurs in apiaries. This also contributes to the persistence of these colonies. Honeybee mating system and population structure and natural selection. In eusocial insects, there is a strong selection for increased genotypic diversity in worker offspring to either meet the demands of different tasks or to mitigate the effects of parasitism. Although all workers in a bee colony stem from a single mother, i.e. the queen, genetic variation among workers can be increased by two mechanisms. One of them is to increase the rate of recombination, the other is by polyandry. Indeed, the rate of recombination in honeybees is among the highest measured in the animal kingdom, and honeybee queens are extremely promiscuous. There is a strong evidence that the genetic diversity that a polyandrous queen generates in her colony benefits its members by enhancing workforce productivity. As honeybees have single locus sex determination, the high genetic diversity is also important to prevent homozygosity in the sex alleles, which results in non-viable diploid males instead of worker bees or queens. Honeybee queens mate in flight with 7 to 28 drones, which may originate from colonies up to 15 kilometers away. Young virgin queens leave the colony for a mating flight. In honeybees, the mating system is characterized by drone congregation areas, DCAs, that are visited by males from many colonies. Baudry et al. Estimated that the drone aggregation area they studied attracted drones from 238 different colonies. Jaffe et al. Found lower numbers in a study of Apis mellifera scutellata in South Africa. The number of colonies from which drones arrive in a particular DCA depend on the density of colonies in the area and on the presence or absence of physical barriers, e.g. bodies of water or mountains. Given the high promiscuity of queens, the long distances travelled by queens and drones and the large number of colonies from which drones in a DCA originate, the conclusion must be that honeybees have a panmictic population structure. This mating structure is important for how natural selection or selective breeding for Varroa resistance proceeds when queens are allowed to mate freely. While selective breeding and natural selection favour resistant traits, mating of young queens with drones from surrounding populations counteracts the effects of local selection. Natural selection for Varroa resistance, the evidence. As documented in the first part of this review, Apis mellifera possesses a number of heritable traits that contribute to its resistance against Varroa. Given the high mortality that Varroa inflicts on non-resistant colonies, it is expected that natural selection upon invasion by Varroa would quickly select for increased frequencies of the resistant traits. Resistance of Apis mellifera scutellata and Apis mellifera campensis in South Africa. Varroa was discovered in South Africa in 1997, where it was most likely introduced with a commercial transport of bees and queens. Two subspecies of honeybee are found in South Africa: the Cape honeybee, Apis mellifera campensis, a coastal race occurring in the fynbos biome along the southwest and south coast of South Africa, and the savanna honeybee, Apis mellifera scutellata, in the rest of South Africa. The mite spread rapidly and after 5 years was found throughout the country. During peak infestations, on average 10,000 to 17,000 mites could be found in a single colony, and sometimes even 30,000 to 50,000 mites. Evidently, there was no immediate impediment to Varroa mite reproduction in Africa Cape and Savannah honeybee colonies, and the mite was able to reproduce very efficiently in both bee subspecies, at least initially. At the peak of the infestation, 30% of colonies collapsed. There was, however, no population-wide collapse of colonies, and the majority survived. The tolerance of Cape and Savannah honeybees for higher infestation rates is likely to be due to the absence of deleterious virus outbreaks, e.g. by a virulent form of deformed wing virus in the South African bees. Although a number of bee viruses have been found in South and East Africa, and Cape honeybee pupae in adults were found to be susceptible to virus infections, it was not possible to induce any bee viruses from Cape honeybee colonies, suggesting a general absence of virulent bee viruses in this population. After the peak infestation, mite densities gradually decreased and Cape honeybees, Apis mellifera campensis, became resistant 3 to 5 years after the arrival of Varroa, while Savannah honeybees, Apis mellifera scutellata, became resistant after 6 to 7 years. Today, Varroa is no longer a problem in South Africa. In Mike Alsop's words, now it is no more than an arbitrary presence. Important for the evolution of resistance against Varroa in South Africa was that the original recommendation given to beekeepers that no chemical treatment should be used until it had been ascertained that Varroa would result in honeybee colony collapse. An additional reason why natural selection could work swiftly was the presence of a large wild honeybee population. Hence, natural selection could operate in both commercial and wild bees, unless hampered by the widespread use of acaricides. As only a relatively minor part of the bee population collapsed, mortality by Varroa did not cause a genetic bottleneck that would otherwise have hampered the evolution of resistance. The following traits have been invoked to explain the resistance. 1. Hygienic behavior. Fries and Rayner report that 77% of pin-killed brood is removed by Apis mellifera scutellata in 24 hours, and a removal rate much higher than reported for European bees. 2. Grooming, measured as a percentage of damaged fallen mites. 3. Non-reproduction. Reproductive failure has increased over time, suggesting that VSH and/or a brood effect has increased over time. 4. Short developmental time. Alsop concluded that the shorter post-capping stage between 9.6 and 12 days in Cape honeybees, 10 to 12 days in Apis mellifera scutellata can reduce reproductive output of Varroa. VSH has never properly been measured in South Africa, but is an important trait in the resistance of Kenyan population of Apis mellifera scutellata. Moreover, it plays an important role in Africanized bees in South America. The most plausible way to explain the changes in abundance of Varroa after its arrival in South Africa is that the frequency of resistance genes in the bee population of Apis mellifera scutellata and Apis mellifera capensis were too low to prevent the observed population explosion, but that most colonies survived peak densities of Varroa because they could tolerate high densities in the absence of virulent viruses that otherwise would have destroyed the colonies. Natural selection for resistance against the mite subsequently took over, resulting in an overall low Varroa density. It is likely that the alleles for the resistant traits, i.e. hygienic behavior and grooming, were already present at frequencies higher than found in European honeybees before Varroa colonized Africa, as Africanized bees showed these behaviors when Varroa arrived in South America. Resistance of Africanized bees. In 1957, African Apis mellifera scutellata bees imported from Tanzania into Brazil to improve honey production in tropical South America escaped from an experimental apiary and hybridized with European bees. The hybrid spread in South America and colonized Central America and the southern United States. Although African honeybees came to Brazil long before Varroa colonized Africa, and before the African Apis mellifera scutellata bees had developed resistance against the mite, the Apis mellifera scutellata hybrids showed already tolerance or resistance to Varroa. Soon after the discovery of Varroa in 1979, the levels of infestation detected were a source of concern for Brazilian apiculture, although there were no reports of bee colony deaths. It soon became clear that Africanized bees can survive Varroa infestations without treatment. Rapid natural selection seems to have resulted in increased resistance, and treatment against Varroa is generally not practiced. Losses of Africanized honeybee colonies due to varrosis are not reported, and possible negative effects on honey production seem to be negligible. This is surprising as, in contrast to South Africa, viruses associated with Varroa-like deformed wing virus are widespread in South America, and Africanized bees are not resistant against the virus, although in one study, the rate of virus increase was lower in Africanized bees than in European bees. The Africanized bee is the common race of honeybee in Brazil. An important prerequisite for the rapid evolution of Varroa resistance was the enormous number of feral colonies of Africanized honeybees in Brazil. Even in natural rainforest ecosystems without any beekeeping activities, the honeybee is the predominant pollinator. Obviously, colonies managed by beekeepers represent only a small percentage of Brazil's honeybee population. Therefore, the feral honeybee population is permanently exposed to selection for Varroa resistance. In Mexico, the Africanized honeybee was established for the first time in 1992 and was found to be resistant against Varroa already in 1994. Thus, it appears unlikely that the resistance evolved there, and more likely that the bees invading from Brazil were already resistant. 1. Hygienic behavior. Africanized bees are generally regarded as having better hygienic behavior than European bees. Reference 51, reference 68, reference 70 found only slightly better hygienic behavior in Africanized bees, most likely because they worked with artificially recapped cells. 2. VSH, reference 46, 171, 172, and provide experimental evidence that Africanized bees have a higher rate of VSH behavior than European bees. Although the first two publications precede Harris's publication in which VSH was defined, the behavior described in these papers is clearly VSH. 3. Grooming. Africanized bees are more efficient in removing phoretic Varroa by grooming than European honeybees. 4. Non-reproduction. Varroa shows a higher rate of non-reproduction in the Africanized honeybee from Brazil and Mexico than in European honeybees. 5. Shorter developmental time. Post-capping period 11.5 to 11.6 days in Africanized honeybees compared to 11.6 to 12 days, in European honeybees. Survival of feral honeybees in Arnott Forest in New York. Varroa arrived in the US in the mid-1980s. A feral population of bees breeding in hollow trees had been censused in 1978, prior to the introduction of Varroa destructor to North America. The census was repeated in 2002 when Varroa had established itself. The number of bee colonies in the forest had not changed. Swarms from this feral population were trapped in the forest and placed in hives to study if the colonies suppressed the Varroa mites. In 2005, no differences in mite population growth were found between the feral bees and commercial non-resistant Apis mellifera carnica bees. It is possible that the relatively short period of time during which the bees of the Arnott Forest had been exposed to Varroa is the reason that alleles for resistant traits had not yet increased, but that the bees persisted because of small colony sizes, frequent swarming, and the widely spaced colonies. An untested additional hypothesis is that the mites and/or the viruses vectored by them may have become less virulent. When colonies are widely separated, their parasites and pathogens are probably transmitted mostly vertically, from parent colony to offspring colony through swarming, a scenario that selects for decreased virulence. Another possibility is that the Arnott Forest bees were Varroa tolerant because they had evolved tolerance or resistance against the associated viruses, as was found for a Swedish population. 10 years later, genomic data showed that major genetic changes had occurred since 1978, and convincing evidence for Varroa resistance in the Arnott Forest honeybee population was found. The feral bees from Arnott Forest are able to reduce mite populations by increased grooming and VSH behavior. Selection for resistance in Primorsky bees. European settlers took Apis mellifera, SPP, Corsica, and Carnica in 1865 to far eastern Russia, Primorsky. The area has native Apis cerana infested with Varroa destructor, which most likely infested the arriving Apis mellifera, resulting in the longest association of Apis mellifera and Varroa destructor. Preliminary examinations of Apis mellifera in the Primorsky territory suggested that they might have substantial levels of mite resistance. These observations inspired the importation of 362 pre-selected queens into North America from 1997 to 2002 for further testing of these Primorsky honeybee queens and the start of a selection program in the USA for Varroa resistance by colony-level selection. An initial evaluation indicated that their commercial traits such as honey production were similar to those of existing commercial stocks. Most importantly, some of the imported Primorsky queens produced colonies which appeared to be resistant to Varroa destructor. After 8 years of selection, several Primorsky queen lines show mite population growth, less than 1, and thus were Varroa resistant. The other lines had growing Varroa populations, and some of them did not much better than commercial Italian bees. Further selection decreased the variance between lines and resulted in overall resistance in the Russian bees. The resistant Primorsky bees exhibited strong grooming traits, high hygienic behavior, reduced breed attractiveness for Varroa, and decreased reproductive success of Varroa in combs built by the Russian honeybees. Unlike Italian colonies, they either slow down or completely stop brood production in response to a lack of nectar flow. This resource sensitivity may also contribute to Russian honeybees' Varroa resistance by interrupting Varroa reproduction. Hence, natural selection in eastern Russia had resulted in a high but variable presence of resistant traits. Artificial selection at colony level in the US, in isolated mating yards, resulted in fully resistant Russian bees. The use of isolated mating yards has prevented the loss of resistant alleles by the dilution effect of the panmictic mating structure, and a well-designed breeding schedule prevented the loss of genetic variation. Resistance in European bees. Unlike Apis mellifera scutellata and Apis mellifera campensis in Africa, and Apis mellifera scutellata hybrids in South America, European subspecies of bees suffered massive colony collapse upon the arrival of Varroa from Russia. The new parasite devastated natural populations of Apis mellifera in Europe and feral populations in North America, and beekeepers experienced massive mortality of colonies. They had no other choice than using acaricides, organic acids, or essential oils to kill the mites, thus hampering natural selection for resistance. Other apicultural practices that are unfavorable for the evolution of Varroa resistance are crowding together of colonies in apiaries so that horizontal transmission of Varroa is favored, managing colonies to be unnaturally large so that they have high honey production and low swarming rates, moving colonies from place to place so that there is strong gene flow that prevents natural selection from altering locally adaptive allele frequencies in a closed population, and regular requeening of colonies with purebred, non-resistant queens. All these practices contribute to making apiaries an ideal environment for Varroa mites and the viruses they vector. In Europe and North America, resistant traits are present, albeit at low frequency in the population, and their expression inside colonies is reduced by the bees in a colony that do not have the resistant alleles. The presence of resistant alleles has been shown by efforts to select for increased hygienic behavior, grooming, VSH, and suppress mite reproduction. However, the expression of resistant alleles in the European honeybee populations is not strong enough to prevent the Varroa population to grow and to prevent colonies from collapse. Natural populations of Apis mellifera and the pollination afforded by them have largely been eliminated by the mite in Europe, threatening the majority of the 10 European subspecies with extinction, and making that natural selection for Varroa resistance cannot proceed in populations not submitted to apicultural practices. Yet, a number of European studies discovered colonies and populations of honeybees that survived the invasion of Varroa without treatment against the mites. Surviving bees were found in Yugoslavia. Starting with 3 colonies that survived an episodic of Varroa destructor, a stock of honeybees was produced which was only slightly more resistant than other stocks. Surviving populations were also found in France and in Norway. Other studies report the importation of bees known to be already Varroa resistant and their survival without treatment after importation and hybridization with local bees. Apis mellifera intermissa imported from Tunisia and surviving bees from Gotland, Sweden, for one of their populations. In addition, there is one well-documented large-scale experiment with a population of untreated colonies kept without treatment. These studies claim that sometimes local conditions allow the evolution of Varroa resistance by natural selection. In the following, we will explore why evidence for the evolution of resistance in Varroa in Europe is rather scanty. Fries et al. Found a genetically diverse honeybee population of 150 colonies on a peninsula at the southern tip of the island Gotland in the Baltic Sea, isolated from the main island through a narrow land bridge. Swarms produced were added as new colonies to the population. After 4 years, 38 new colonies had been established from swarms, but mortality due to Varroa infestation resulted in only 13 colonies of the 188 surviving after 4 years. The colonies surviving after 4 years had mite infestations that were more than halved in comparison with the 3rd year before massive colony collapse occurred. In addition, surviving colonies had fewer worker bees and produced fewer drones. The small number of colonies surviving resulted in a genetic bottleneck and strong inbreeding. The experiment shows that genetic variation for resistance was present in the population before selection, and that natural selection to improve Varroa resistance is possible in closed populations, albeit at a price of lower brood production. LeConte et al. Allowed 82 colonies that had survived the invasion by Varroa without treatment. They were placed in the region where they had been found, 30 in an apiary near Le Mans and 52 in an apiary in Avignon. Treatment control colonies were placed nearby. The mortality of colonies varied between 9.7 and 16.8% per year. Surviving colonies had only 32.4% of the Varroa infestation rate in control colonies. Honey production by untreated surviving colonies was half of that of the controls. The experiment showed that the collected colonies had some degree of Varroa resistance at the start of the study. The surviving colonies were maintained under artificial selection, as is witnessed by this citation. Open quotation. What has happened to these bees since we published those results in 2007? Once every 2 years, we graft queen larvae from the 3 best colonies in each apiary, west and south of France, to get 20 colonies. The queens are naturally mated by local drones. About 30 to 35% of the colonies die within 18 months, but the rest of the colonies are good candidates for surviving to the mite, so the stock still survives efficiently. Close quotation. Hence, despite continued selection, Varroa resistance has not increased over a 10-year period. The natural and artificial selection for colony survival did not increase the frequency of resistant genes, probably because the bees are kept in an open panmictic breeding population. While selection favours colonies with a higher frequency of resistant alleles, panmictic mating in a population with a low frequency of these alleles makes the queens of the selected colonies mate with drones with a low frequency of the resistance alleles. In addition, drones with resistant genes from the untreated colonies disperse into the environment. Thus, the mating structure of the population counteracts local selection for Varroa resistance. Colonies descending from both the Avignon and Gotland survivor populations both still harboured growing mite populations with more than 0.7 fertile female offspring per foundress per cycle, and would have collapsed if left to natural selection alone. They survived because they were subjected to prolonged artificial selection and periodically multiplied by breeding a large number of queens from the best colonies. When the Avignon bees were tested outside their native environment in a European-wide experiment, neither their Varroa infestation rate after 1 year without treatment nor their survival outperformed that of colonies descending from non-selected genotypes tested at the same locations. The surviving colonies from the Auslandet region in Norway were part of an open panmictic breeding population, the population being within sufficient distance of known susceptible colonies from various backgrounds, mostly Apis mellifera mellifera and Apis mellifera carnica, buckfast, that would facilitate horizontal parasite transfer. These colonies were also multiplied by splitting the healthy surviving ones to replace lost colonies, and hence they were also under a continuing regime of artificial selection. The surviving colonies harbouring growing mite populations, 0.87 fertile female offspring per foundress. Which mechanisms are involved in producing mite reproduction in these populations? The role of hygienic behavior was investigated in the surviving Gotland colonies by studying the fate of 100 pin-killed pupae. Removal rate in 12 hours was only 15%, and not different from the 20% found in control colonies, not supporting a role for hygienic behavior. Unfortunately, the authors did not test for VSH behavior, leaving the possibility that the removal of mite-infested pupae plays a role in reducing Varroa reproduction. They also measured the percentage of damaged mites of mites fallen onto the bottom board. As we have seen above, damaged mites can both result from VSH behavior as well as from grooming behavior. They found 31 to 35% of damaged mites, which was not different from the percentage found in unrelated control colonies. Although the results are not supporting the hypothesis that grooming plays a role in the reduction of the Varroa population, they cannot be taken as evidence that grooming plays no role. Experiments using a better assay to assess grooming, e.g. that of reference 78, are needed. The surviving Gotland colonies had a significantly lower proportion of mites that reproduced successfully, 48% versus 78% in control colonies. Failure to reproduce resulted from the infertility, absence of male offspring, high proportion of mite offspring mortality, or delayed egg laying by the mother mite. As we have seen, there are 4 possible mechanisms to explain an increased percentage of non-reproduction in Varroa, 2 of which caused by the bee, i.e. VSH behavior and brood effect. Proper experiments on VSH behavior and a brood effect are needed to distinguish between these hypotheses. As the resistant bee populations originated from only 13 surviving colonies, it is possible that traits like VSH and grooming were not present in the small sample. Locke and Fries suggested that the smaller colony size of surviving bees is an adaptation that would reduce reproductive rate of Varroa. As smaller colony size decreases colony fitness in many ways, we prefer the alternative hypothesis that the smaller colony size is non-adaptive and an effect of inbreeding in this population. They also suggested that Gotland bees were resistant to viruses that normally cause colony collapse. This was confirmed by reference 130 to 132 and reference 133. Why did the Gotland experiment not result in fully resistant bees? As only 13 of 188 colonies survived, and genetic drift had caused an extreme loss of genetic diversity in the surviving population, it seems likely that the insufficient genetic variation hampers the evolution to full resistance in this population. This hypothesis should be tested by increasing the genetic variation in this population and document the changes in mite reproductive success. The Varroa mites in colonies of the Avignon population of surviving bees also had a lower proportion of reproducing mites, with non-reproduction being the most important factor. The mechanism causing the reduction in reproduction was not determined. VSH could possibly explain the suppressed mite reproduction in this population, as genetic evidence suggests that the suppressed mite reproduction is caused by a behavioral trait. The mechanism of resistance in the Le Mans population has not been studied. Reduced reproductive success of Varroa was also observed in the surviving colonies from the Østlandet region in Norway. This can be completely explained by VSH behavior, as the frequency of VSH was only 5%. Audi et al. Suggested that a slightly shorter post-capping period for the brood of surviving colonies in comparison with non-related controls could have contributed to the lower Varroa reproductive rate, but did not calculate whether this small difference could produce the observed effect on mite reproduction, which seems likely. Other mechanisms, like a brood effect, were not studied. Kifas started Varroa-resistant lines by importing Apis mellifera intermissa from Tunisia. Judging from the very high rates, 40 to 75%, of non-reproduction in these bees, they had already a high incidence of VSH behavior when arriving in France. Evidence for high rates of VSH and grooming in these bees is provided by reference 62, reference 201. In a Dutch experiment on survival of untreated bee colonies, Panziera et al. Found evidence for the role of VSH in reducing mite populations. None of the European bees cited above have resulted in a fully Varroa-resistant population. This is either because surviving colonies were part of a paramictic population and surrounded by colonies with low frequency of resistant traits, or, in the only example of closed population, Gotland, because of inbreeding. Conclusions. The host shift of Varroa destructor to Apis mellifera and the fast colonization of Apis mellifera populations in Europe, the Americas, and Africa initially resulted in considerable mortality, exerting strong selection for the evolution of resistance against the mites. While selection resulted in resistance in populations of Apis mellifera scutellata and Apis mellifera capensis in Africa, and in Apis mellifera scutellata hybrids in South America, resistance did not evolve in honeybees in Europe and North America. Most unprotected colonies of European honeybees in these continents succumb when infested with Varroa, and resistance to Varroa has not increased in the 40 years after the Varroa invasion. We have reviewed research on traits for Varroa resistance and the literature that documents the evolution of resistance to Varroa in Africa and in South America. We have also reviewed studies of several populations on honeybees in Europe and North America that were not treated against Varroa, and the evidence these studies provide for natural selection of Varroa resistance. From reviewing this evidence, we conclude that 1. Natural selection can result in population-wide resistance in large palmictic populations only when a large proportion of the population survives the initial invasion of Varroa. This is what happened in Africa and South America. 2. When upon invasion of Varroa, a major part of the bee population collapses or is protected against the mite by chemical treatment, natural selection for resistance does not succeed. This is what occurred in Europe and North America. 3. Small colony size, frequent swarming, and widely spaced colonies of wild or feral honeybees reduce the risk of colony collapse due to Varroa infestation. These life history and ecological characters promote vertical transmission of mites, and viruses are therefore conducive to the evolution of less virulent mites and viruses and more resistant bees. These characters also allow the evolution of resistance in wild bee populations. This is shown by the study of bees in the Arnott forest. 4. The palmictic mating structure of many honeybees prevents local natural selection for resistance, because resistance genes disperse into neighbouring populations at a rate higher or equal to the local rate of recruitment of these genes by selection. 5. Selection by breeding can increase the level of resistance of colonies and so increase the proportion of resistant colonies in the population as a whole. When this proportion is high enough, beekeepers can stop chemical treatment and natural selection can proceed. 6. In closed populations, as on islands, natural selection is not counteracted by the dispersal of resistant genes, and natural selection can proceed unless it is constrained by inbreeding. This is what the Gotland experiment has shown. In most of the populations that develop resistance against Varroa, behavioural defences against the mites are important. Grooming against phoretic mites and hygienic behavior, or more precisely, VSH against reproducing mites. In addition, brood effects and shorter developmental times play a role in reducing Varroa mite reproductive success. The exception is the Gotland experiment, in which no evidence was found for grooming or hygienic behavior. On the whole, these results show that natural selection favours traits that also have been used in selective breeding programs. The ultimate goal of making European and North American honeybees resistant against Varroa is within reach. Artificial selection using single drone insemination, as pioneered by reference 32 and reference 86, can be used to increase the frequency of resistant alleles in the honeybee populations of both continents. Natural and artificial selection at colony level can also be used in closed populations, e.g. on the islands or otherwise isolated mating yards, providing that genetic variation in these populations is maintained. Resistant colonies produced in this way can then be used to increase the level of resistance in large palmictic populations. Once the resistance level has passed the threshold where it becomes profitable for apiculturists to stop chemical treatments of the mite, natural selection can proceed to make European and North American honeybees fully Varroa resistant.